Welcome back. What we're going to do in this video is talk about binding energy. Okay, and binding energy. Okay, so if we have a diagram, okay, so here's our free energy on the y-axis and our reaction coordinate is on the x-axis. Let's say we have a hypothetical exergonic reaction. Okay, negative delta G, right? So we have that, and then let's say that's non-catalyzed, right? And again, we have our transition state up here, right? But let's say we catalyze it and we break it up into mechanistic steps, right? Okay, like that. And again, we have a transition state for each of these, right? Okay. And effectively what binding energy is, okay, is it's the difference in energy between the highest transition state in each one of these steps, both catalyzed and non-catalyzed. So in this case, it's basically this right here, okay? So this is your binding energy, and we typically put that as delta G B, okay, for binding energy, okay? Now, it turns out that delta G of binding is actually not very useful, okay? At least for an undergraduate understanding, it's not entirely useful, but the concepts that come out of it are, okay? And there's three really big concepts about enzymes that come out of binding energy, okay? And so two of them have to do with entropy. The other one is called induced fit, okay? And you've probably heard of induced fit when you've talked to in maybe upper level biologies and so forth. But first we're going to talk about entropy, okay? Now, the first part of entropy is an entropy reduction, okay? So if we have an entropy reduction, that implies that we have a negative delta S. Recall that entropy is itself a state function, meaning that it only depends on the final and initial states, not on the path that it takes to get to the final state from the initial state. Okay, so it's a state function. And one of the good things that we'll talk about when we do calculations with it is you can actually add delta S's, just like you can add delta G's for various reactions to get a net delta G. You can actually add delta S's as well. Okay, so let's talk about what happens when you have an entropy reduction. Okay, and to understand it, let's look at a hypothetical, actually not a hypothetical, it's a real reaction. Okay, let's say these are the walls of my active site. Okay, now this example that I'm using, okay, this is going to be the active site of hexokinase. Hexokinase, we talked about it in the cofactor video, okay, is ultimately the enzyme that's the first reaction in glycolysis. It catalyzes that reaction. And what you would remember if you watched that video is that this molecule binds an incoming ATP using the chelate effect from a magnesium ion that on one side chelates the incoming ATP, and on the other side it's chelated by the aspartate residues in the active site. Now, whenever you see this term ado, that just means adenosine. So I'm denoting that this is adenosine triphosphate. Okay, so what you have is you have this interaction on one side between the magnesium and the aspartate residues, and then on the other side you have the magnesium interacting with the phosphates. Okay, and typically it's just the beta and the gamma phosphate. Okay, now recall that phosphate's counter ion in the cell is magnesium. So if you were to look at DNA that has lots of phosphate on it, you see your DNA is encompassed by a bunch of magnesium ions. Okay, so all phosphates accompany magnesium. Now you can imagine that that if I have a magnesium ion that's chelating the incoming ATP, you can imagine that's a very stabilizing effect. Okay, and in general, negative delta S's correspond with stabilizing, where you're going to a more stable state. Okay, so let's think, for instance, what happens if I don't have this magnesium, okay? If I don't have this magnesium, right, what happens to the ATP? Well, it might enter the active site, but it's going to be flopping all around, right? What the magnesium is doing is it's holding that ATP in place, right? So in other words, what's happening is there is a decreased number of positions that ATP can be in, right? So in other words, the magnesium is just holding the ATP in place through electrostatic interactions, right? Magnesium has a 2+, ATP has negative charges, right? So effectively, magnesium is holding that ATP in place, right? Now, one important thing that comes out of this is one of the formulas for entropy that was discovered by a gentleman named Ludwig Boltzmann, okay? And he derived an equation that basically says that entropy for a particular molecule is equal to Boltzmann's constant times the log of the number of microstates. And W here is the number of microstate. Now, what is a microstate? A microstate is a position that a molecule can be in. Okay, so if we define a volume, okay, an area of space, right? In other words, the area of space could be our active site. Now, in reality, you have an unlimited amount of volume because you have an unlimited area of space in the universe. I mean, we go out of the active site and go into the bounds of outer space, right? But we're just concerned with the active site, right? So we have a fixed volume, and there's going to be a corresponding number of microstates that can be in that volume in the active site. Now, you can imagine if that magnesium wasn't there, what happens? That ATP is flopping all over the place, right? So it has a lot of microstates. What happens to the number of microstates if I put a magnesium in there? The magnesium holds that ATP in place, so the number of microstates, the number of positions that ATP can be in, is minimized. So what happens to S, the entropy? It drops, right? So what you have is you have ultimately a decrease in the entropy when you have weak interactions. What's a weak interaction? So a weak interaction, okay, we have weak interactions, okay? These are things that are going to be non-covalent, okay? Non-covalent bonds. So these are things like electrostatic interactions between full positives and full negatives, or like in this case, or we can have things like partial charges that interact, like hydrogen bonding, dipole interactions. Basically, any one of your intermolecular forces that you learned in general chemistry, those are going to be your weak interactions, okay? So what it turns out is that weak interactions in the active site between the substrate and the enzyme are stabilizing, okay? And because magnesium is a cofactor, we're considering it part of the hollow enzyme, okay? Okay, hollow enzyme, not hollow as in like a hole, I mean like hollow, H-O-L-O enzyme, okay? So magnesium is part of the hollow enzyme, right? In other words, it's critical for hexokinase's function. Without magnesium, you can't bind ATP, or at least it can go into the active site, but there's not enough, there's too many microstates, there's too much entropy for the catalysis to occur, right? Now, effectively what I'm doing by having this magnesium in the active site is I'm stabilizing the ATP. Now, it turns out that what happens is there's a nucleophilic attack of a hydroxyl group of glucose, so that you have a glucose molecule that has a hydroxyl group, and effectively what happens is there's a nucleophilic attack on the gamma phosphorus, right? And we're not going to go into the whole mechanism here, but the important thing is to understand is that glucose does a nucleophilic attack on the gamma phosphate. Now, here's an important analogy that we can have with respect to this enzyme, okay? We know magnesium is holding ATP in the active site, right? Now, let me ask you this. Have you ever seen those movies where you got like these mobsters, right? And they're beating up someone, right? And they've got this guy who's being held in position by two mobsters, and then another mobster is kind of punching him in the stomach, right? Okay? Is it easier to punch the guy in the stomach when you've got two guys holding him in place, or when he's running free away from you, right? Well, it's easier to punch the guy when you're holding him in place, right? So just like that, what happens on the macro scale happens on the micro scale, right? So it turns out that it's much easier to do the nucleophilic attack on that phosphorus if you're holding the ATP in place. In other words, if you have that entropy reduction, you have lower number of microstates, you have lower entropy. So it's much easier to do that nucleophilic attack when you're holding the ATP in place. So that's the important thing that comes out of an entropy reduction. You're holding it in place, and that holding in place occurs due to weak interactions. Let's think about another example. Let's say that I have the walls of an active site right here. Okay. let's say I have a molecule. Let me think of a good molecule. Let's say I have something like this. Let's say I have this, and again, it wouldn't be like this at physiological pH, right? But for the purpose here, just think about it like this, okay? Now you have to imagine there's a partial positive on the hydrogen, partial negative here, and a partial negative here, right? So we've got to think about what kind of residues might interact with these things, right? And we're assuming that it's protonated, right? In reality, we know it's not protonated, right? But we're just going to think of it like that for now, okay? Now, what kind of things might interact with a partial positive of a hydrogen? What kind of things might interact? Well, what kind of residues interact with a partial positive? Well, they'd be negative, right? So what's one like that? Might be glutamate, right? Glutamate has a full negative, right? You can imagine that partial positive is going to want to interact with that partial positive. As I say, the partial positive is going to interact with that negative charge, right? Now you also have this partial negative of this oxygen. What kind of things might want to interact with a partial negative? Well, you can have a lysine, right? 1, 2, 3, 4, right? You might have a lysine, right? That positive on the lysine can interact with the negative of the oxygen in that carbonyl, right? So can you see now why understanding why these amino acids have charges is so important? The charges are important when we get into the active site of an enzyme. They help a lot of times stabilize an incoming molecule that's coming into the active site. So very commonly, you'll have carboxylates that stabilize things with partial positives that come into the active site, or sometimes full positives, right? Other times, things that have partial negatives or full negatives will be stabilized by things like lysine or arginine. So things that would be stabilized by, or things that stabilize a partial negative might be lysine or arginine, right? Things that have full positives, right? And things that might stabilize an incoming positive are going to be things like glutamate and aspartate. Right? Those are things that might stabilize an incoming positive charge, right? And the reason is because you have a dipole moment, right, towards the oxygen, okay? So these are weak interactions, and these weak interactions help stabilize incoming molecules into the active site, right? And so in other words, what you're doing is you're stabilizing it by holding this molecule in position so that you can easily get a nucleophilic attack, right? If you have this molecule held in position, the enzyme, what the enzyme is designed to do is it's designed to hold something in proper orientation, okay? And as we'll find later, a lot of times what happens is the enzyme is specifically made to hold a molecule in a proper position so that another residue can do a reaction, okay? But for now, we're just worrying about the weak interactions and how they stabilize the molecule. So if you stabilize this carboxylic acid in the active site, okay, you're decreasing the number of microstates and therefore decreasing the entropy, okay? So it's an overall decrease in entropy. Now, that may seem like an overall unfavorable thing, but it turns out there's something to compensate for that, and that's called desolvation, okay? Let's go over here to talk about desolvation. Now, again, we have the walls of the active site, right? And let's do our example before. Let's say 1, 2, 3, 4. Let's say we have a lysine, right? Here's a lysine residue. Let's say we also have a glutamate. 1, 2, 3. So we have a glutamate, right? Now, if I have a water molecule, here's my water molecule, okay? Okay? And there's a reason I drew it like this. If you notice, the hydrogen has a partial positive and the oxygen has a partial negative, okay? And you would see this throughout the active site, okay? So what you have is that the partial negative of the oxygen interacts with the lysine, or you could have arginine or something like that. And the partial negative is going to interact with the partial positive of the hydrogen. These are just opposite charges attract, okay? And so the water molecule is going to be interacting with the residues in the active site. Now, what you have to understand is that the protein is designed to very, very powerfully and strongly interact with its substrate. So if you could basically think that it's more thermodynamically favorable for the enzyme to interact with the substrate than it actually is to interact with water, okay? So you have a substrate, okay, so here's my substrate. Here's my substrate, and it's going to come into the active site, okay? Now, it's more thermodynamically favorable for the substrate to interact with the residues in the active site. And so you have to imagine there's all sorts of waters in here, right? Not just that one. There's all sorts of waters, right? Okay. So when the substrate comes into the active site, okay, you have to imagine that all these waters are going to be expelled, right? So out of the active site, you have some amount of waters, right? N water. I'm just putting N because, again, we don't know how many waters it is, but it's a good number of them, right? So when the substrate comes in, it's thermodynamically more favorable for the substrate to interact with the residues than it is for water. So the water gets expelled from the active site. Now you can imagine that even though there's a decrease in entropy with the substrate binding due to weak interactions, now you have an increased number of microstates for the water. Right? The water gets expelled. Now there's an increased number of microstates. So if I have my equation, the entropy is equal to Boltzmann's constant times the log of the number of microstates. If I increase the number of microstates, what happens to the entropy? It increases, right? So now you have an increase in entropy that's associated with water being expelled from the active site. And we call this process desolvation. Okay? Desolvation is basically, you can think of it as an expulsion of water that occurs when the substrate binds in the active site. Okay? And this process gets a positive delta S. Okay? And effectively what happens is this positive delta S more than compensates for the negative delta S that we saw over here. Okay? So overall, the process has a has a positive delta S. And if you think about it like this, right, your delta G of reaction, okay, is equal to delta H minus T delta S, right? So if you have an overall, if you add those two delta S's, right, if you overall have a positive delta S, then assuming your enthalpy is negative, you overall have a negative delta G. And that comes from a positive delta S, right? So positive delta S contributes to the release of free energy, right? So overall, because you have this desolvation, that more than compensates for the entropy reduction when you bind the substrate, okay? So you have an entropy reduction when the substrate binds, but also when the substrate binds, water gets expelled, there's an increased number of microstates, and the entropy ultimately increases, okay? So overall, there's an increase in entropy, okay? And all this has to do with these weak interactions between the enzyme slash coenzyme slash cofactor and the substrate, okay? So again, this is all caused by weak interactions. Positive wants to interact with positive, or excuse me, sorry about that. Positive wants to interact with negative. Negative wants to interact with positive. And if you have two hydrophobic molecules, okay, those hydrophobic molecules are going to interact together because hydrophobic doesn't want to interact with hydrophilic. Okay, so if you have something like this, let me draw a hypothetical molecule. Okay, what I'm drawing now is a very messy structure of cholesterol. Okay, so you have something that looks like this. And you can already see that it's a very large carbon chain, right? Now, if I asked you that is very messy. If I asked you which residues might want to interact with this, what would you say? Well, you'd probably say something like phenylalanine, or tyrosine, or tryptophan, valine, leucine, isoleucine, right? Hydrophobic residues want to interact with hydrophobic substrates. So if you have an enzyme like cholesterol oxidase or the cholesterol side chain cleavage enzyme, I'm more than willing to bet you would find some of these that help bind cholesterol. So if you have a hydrophobic molecule, it's going to be bound by hydrophobic substrates, okay? And also, if you have a polar molecule, it's going to be bound by polar substrate. Or polar substrate is going to be bound by polar residues, okay? So negative interacts with positive, positive interacts with negative, okay? Excuse me. Okay. So I hope this makes sense, okay? And again, all this entropy stuff deals with weak interactions. Now, the last part that has to do with binding energy has to do with changes in conformation, and that has to do with induced fit, okay? Now, what you have to think about is when you have things in the active site, okay, the enzyme changes conformation. So one thing I would have hoped you get out of biochemistry is that anytime something binds to a protein, it changes conformation. It can be something as minuscule as a proton, and it doesn't even have to be a bond. It could just be an intramolecular force. Anytime you have that, the protein changes conformation. Proteins are dynamic, right? Something binds to an enzyme, it changes conformation. So let's say you have this hypothetical situation, okay? Okay, or actually, let me do this, okay? We're going to use our example here, over here. Where was it? I think it might have been down here. I knew I drew it. Okay, here we go. Okay, let's say, for instance, this is a serine hydrolase. Okay, serine hydrolases, what they do essentially is they use serine as a nucleophile in their mechanism. Usually, what they're doing is they're doing a nucleophilic acyl substitution. Okay, and they're doing it specifically on a carboxylic acid derivative like an ester. Okay, so this would be a serine esterase. Now, let's say, for instance, that you've got these residues that are binding this incoming molecule, right? But you also have this serine residue. Now, if I draw the serine residue like this, okay, serine has a lone pair, right, on the oxygen. Now, if I asked you, is that really close enough proximity to do a nucleophilic attack, what would you say? Well, no. That's not close enough proximity for this hydroxyl group to do a nucleophilic attack right here. It's not close enough. So what ends up happening is when the substrate binds to the active site, remember, it's stabilized in there through amino acid residues and sometimes with cofactors and coenzymes, right? But what ends up happening is the enzyme changes conformation. So let's say, for instance, that let's say you have your hypothetical binding site. Where was it? Okay. So what did I have? Let me go back and look. I don't remember what it was. So I had glutamate and lysine. Okay, and I had ester. Right. So here, go back here. Where am I? Did I even finish drawing it? No, I don't think I did. Okay, let's do this. I have a glutamate, right? So here's my glutamate. Glutamate has a net negative charge. I have 1, 2, 3, 4. Right? I have my lysine, right? So that lysine interacts here, right? This has a partial positive. This has a partial negative. Okay, and then this is just an R group. Okay, so that's that. Now, we originally bound it in the active site, right? Now, remember what happens anytime you bind something in the active site. What happens? There's a change in conformation. If we go back and look at our picture, right, what happened? Was there enough? Well, just notice there's too much distance between the serine and the carboxylic acid derivative, so no nucleophilic attack is going to occur. Now you have to imagine that what happens if you change conformations? Well, something that might happen is you change the conformation such that now the serine is in close proximity to the carboxylic acid derivative. Now you're in close enough proximity, so now you get a nucleophilic acyl substitution. And we've seen this mechanism before. What might happen is you kick off this group or something like that. Okay? So when you change conformation, usually what happens is you bring the substrate in close proximity to catalytic residues. And we'll go into more detail on catalytic residues later. But a lot of times things like serine and cysteine act as nucleophiles. Things like lysine, arginine, glutamate, aspartate can act as Bronsted-Lowry acid-based donors, acceptors, things like that. Okay? But the point is that when the enzyme changes conformation after substrate binding, you have the substrate getting into close proximity to catalytic residues, okay, like a serine nucleophile. Okay, and notice I tried to illustrate it here. The enzyme, the active site wall changed conformation to bring the serine in close proximity. Okay. Now you have to imagine that what I challenge you to do is to pause the video and draw the mechanism of this reaction. And again, this is just a Fischer esterification reaction. Remember those from organic. It's a Fischer esterification. So draw the mechanism for it. And what you would find is that for the mechanism, for every single step in the mechanism, there's a corresponding change in conformation. And those changes in conformation bring certain residues in close proximity to one another, right? I hope that makes sense. So for each mechanistic step in the reaction, there is a corresponding change in conformation. This concept is called induced fit. Okay, so whenever you change conformation, you effectively bring catalytic residues in close proximity, and they can be things like nucleophiles, or they can be things like Lewis acid base. If you have a Lewis base, it can be a nucleophile or something like that. Okay, examples of that would be serine and cysteine. Okay, sometimes even tyrosine can act as a nucleophile. Okay, but the whole point is that changes in conformation bring residues in close proximity with the substrate, and so that is what does the catalysis. And what you might find is maybe, for instance, when you change conformation, maybe this wall of the enzyme would move over there. Or maybe this would come back over here or something like that. Okay, the enzymes are dynamic, so when you change, when you bind something, there is a change in conformation. Okay, and that's really the whole point of this video, is you've got changes in entropy and you have changes in conformation. Okay, so the first concept we talked about, let's do a little recap. The first concept was an entropy reduction. Okay, the entropy reduction occurred when we had the substrate stabilized in the active site. Remember, when you have a negative change in entropy, what happens? Negative change in entropy corresponds to a stabilizing effect. When you stabilize something in the active site, you're effectively restricting its range of motion. You're reducing the number of microstates it can be in, so there's a corresponding drop in entropy. And that drop in entropy, the reduced number of microstates, causes it to be more susceptible to things like nucleophilic attack. Remember, for this ATP hexokinase mechanism, remember our sort of analogy to the mob, right? It's a lot easier to punch someone when you have two guys holding somebody down, right? Versus trying to punch a guy who's running away from you, right? Think about it like that. So entropy reduction. Then we have desolvation. Desolvation forces water out of the active site because it is more thermodynamically favorable to interact with the substrate than it is for water. So when water gets expelled from the active site, there's an increased number of microstates for the water, so there's an increase in entropy. And that increase in entropy more than compensates for the decrease in entropy that we get from substrate binding. Okay, and all of these things are done ultimately through weak interactions. Remember, those weak interactions are electrostatic forces, ion dipole forces, hydrogen bonding, dipole, things like that. Okay, and we can even have things like down to van der Waals forces. Okay, and then the last point I want to make is just that, remember, for every mechanistic step in an organic mechanism, there is a corresponding change in conformation of the enzyme. Remember, enzymes are dynamic proteins. Okay, and this concept has to do with induced fit. So I hope this video gave you a little bit of intuition on what exactly is happening with an enzyme in the active site. So see you in the next video.